Hey judges and everyone watching. We are Group 4 from 10 Linnaeus conducting research under the Life Science category. I am Andre Diaz, the leader of the group, and I have a question for everyone. Has anyone noticed a drastic increase in onion prices?
We all have, and this is due to pest infestation, particularly by the beet onion. This significantly impacts crop production and efficiency, notably in onion cultivation. Bao is highly damaging, targeting over 90 plant species, including onions, lettuce, corn, and potatoes. This is one of the main reasons why the agricultural sector is having a hard time providing cost-friendly crops in the Philippines.
In the past, people have tried combating this issue by using commercially available pesticides. However, these contain harmful chemicals that can affect the quality of the crops, as well as the health of farmers who are exposed to them for long periods of time. They also cost a lot, which leads to the agricultural sector having problems making crop prices lower.
To address this challenge, our research group developed a novel solution: a larvicide made out of Indian rubber vine leaf extract. Did you know that Indian rubber vine is a potential plant that could be turned into a larvicide against beet armyworm? It is also one of Australia's most invasive weeds. It proves abundant and cost-effective, ensuring a sustainable solution to bee infestations on onion crops.
Now, let's discuss the objectives and significance of the study. Starting with the objectives, our study aims to produce a larvicide that is cheaper and safer than existing larvicides while correlating with the efficacy rate. Additionally, we aim to increase the quantity and quality of onion crops produced while using the Indian rubber vine as a larvicide.
This study will develop an accessible, economical, sustainable, and natural solution to pest problems, prevent infestation to increase crop production, and assist farmers in enhancing their supply of onions and augmenting their output. In addition, this study meets the following Sustainable Development Goals: SDG 1, No Poverty; SDG 2, Zero Hunger; SDG 12, Responsible Consumption and Production; and SDG 17, Partnership for the Goals.
So, how was the study conducted? The CLSU Plant Pests and Disease Research Center supplied the beet armyworms via the Nanotechnology Research and Science Facility, while the Indian rubber vine leaves were gathered from Bulacan. The leaves underwent rinsing, air drying, and small piece cutting before being over-dried and grinded. After it was grinded, 900 milliliters of ethanol were added to macerate it. After 3 days, the mixture was filtered to get it ready for rotary evaporation, cruding, dilution, and concentration measurement. This was followed by the bioassay. The phytochemical test was then carried out to determine which phytochemicals were present in the plant and whether they were also present in commercial larvicides. Once the results were released, the extract was analyzed using the FTIR test to check for contamination.
Moving on to the results and discussion, here are the results for our first bioassay, contact toxicity. We found that after 72 hours of exposure, the treatment with 6,000 parts per million had the highest contact mortality rate with 50%. For the probit analysis, we determined that after 72 hours, the beet armyworm mortality increases with exposure.
For our second bioassay, residual toxicity, we found that after 72 hours of exposure, the treatment with 25,000 parts per million had the highest contact mortality rate with 72%. After 72 hours, the probit analysis testing results determined that the beet armyworm mortality increases with exposure time.
The phytochemical analysis and FPIR test identified the presence of saponins, tannins, terpenoids, cardiac glycosides, coumarins, hydroxyl groups, alkanes, and carbonyl groups, which overall suggests that the Indian rubber vine demonstrates larvicidal properties and could be used for future purposes in the future.
We have accomplished a lot in this study. However, we still lack significant concepts in this part. The recommendations for field testing could have been conducted, particularly on onions, to provide insight into its effectiveness in controlled field conditions. Additionally, further research could explore the Indian rubber vine's extract's effects on other pests in the Philippines. Following experimentation and results, branching into nanotechnology research was also considered to enhance larvicide efficacy, durability, and safety.
To end this presentation, our group confidently concludes that Indian rubber vine leaves does have larvicidal potential against the beet armyworm and can be an alternative to commercial herbicides. That is all for our group. Thank you for listening. To note, here are the references for our research. Thank you for listening.